this is indeed going to be an exciting day. For those of you who don't know me, I'm John Soderstrom, the Managing Director of the Office of Cooperative Research, and I want to welcome everybody here. Just so you understand, we have over 1,000 pre-registrants for this meeting today. We had to cut it off. Sorry, folks, but thank you for coming. This is awesome to see everybody here. For those of you who are in the business of starting businesses or interested in those things, we have over 175 investors in the room, maybe not right this moment, but they're going to be here today representing over 60 investment funds. So that is amazing. And I think by Connecticut standards, that's got to be off the charts with anything that's ever happened before. So we are really, really excited about what's going to happen today. As you might understand, with 1,000 people showing up, this is probably the biggest thing that we've ever done on the Yale campus outside of commencement, and my office doesn't handle commencement, so we're not sure about all these things, so just bear with us a little bit. And if you didn't get registered ahead of time because of lines and stuff, trust me, you'll still be able to get back there and do those things. But I just really am very excited that everybody's here. But before I get started, obviously none of these things happen without sponsors. And I don't have control of the podium, so I don't know how to click the slides forward. And somebody's going to tell me how to do that. Okay. I am AV challenged, I admit it. Okay, I want to particularly thank our sponsors. We have some amazing folks who have been with us. And many of them have been sponsoring this for a number of years, and they're part of our lead sponsors. So to Saul Ewing, to Goodwin, to Connecticut Innovations, Wigan and Dana, to Royalty Pharma, Merck, Shipman Goodwin, Wilson Sonsini, as our lead sponsors, we thank you. But we have other sponsors as well. We have lots of sponsors. And so one of the things, you'll see tables and you'll see them around the room, and I really would hope that you would reach out and show your appreciation for all that they've done, because trust me, none of this happens without their support. So it's all good. There are also a couple of people that I want to specifically recognize, because as you might imagine, trying to get organized for something this size, and I have a very small staff, trust me, I'm going to say that multiple times, but there are two people, two human beings who have gone way, 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 way beyond the call of duty, and it's going to be hard for me to recognize them because they're standing way in the back, but I want to recognize Ann Holman and Tim Obstrup from my staff, because they have been amazing. Give it up. So the other thing I want to do is I want to thank the School of Management, because they gave us the right to use this venue. They usually restrict it to just their own people. So, but we want to make sure that we treat it with respect. And so you may or may not have seen the sign when you came in, but no food or coffee. There are water bottles. Those are allowable inside, but otherwise, please don't bring those in. But we want to treat it with respect so we can use this again next year. So thank you for that. And now, because you get tired of hearing from me and you're going to see me more than once today, I want to introduce Michael Crair, who's the Deputy Dean for Scientific Affairs at the School of Medicine, and he's going to introduce our very first speaker today.
Cheers. Thank you, John. It's wonderful to see a full packed audience. It's a great schedule, great set of speakers today, and it's my pleasure to introduce Jonathan Rothberg. So Jonathan is a native of New Haven. He was born and raised in New Haven and lives now along the shoreline just to the east of New Haven. He got his bachelor's degree in chemical engineering from Carnegie Mellon, came back to Yale for his PhD in biology up here on Science Hill. And I'm just going to perseverate for a minute on his PhD because he discovered a very interesting protein, SLIT, which is involved in nervous system development. Actually, it's fundamental in midline guidance of axons across the midline and bilaterally symmetric nervous systems. And actually, the work was done in Drosophila, a fly, and it sort of illustrates the importance of fundamental research in model systems, because those discoveries actually of SLIT and associated proteins turns out to be involved in angiogenesis and actually cancer as well. Following his PhD, Jonathan founded a remarkable set of biotech companies, including let's see, there's a long list. I'm not going to list them all. Curagen, 454 Life Sciences, Ion Torrent, and more recently, the medical device incubator for Catalyzer here in the New Haven area. So these biotech companies are remarkable and notable for their highly inventive technology and also, for instance, next generation sequencing, which was responsible for the complete sequencing of the first human genome, James Watson's sequence, and also, for instance, the discovery of the entire Neanderthal genome more recently. So Jonathan continues to publish scientific articles as well as do invention of biotech and catalyze biotech across the nation. So his inventions have really revolutionized genetics and are in the process of revolutionizing medicine and the treatment of disorders. So he's very widely published, as I said. He's received a numerous number of awards, including the National Medal of Technology and Innovation from President Obama a few years ago. He's elected member of the National Academy of Engineering and a set of other honors, which are quite long and very fitting for his accomplishments. So please join me in welcoming Jonathan Rothberg.
Thanks for the wonderful introduction. I want to start with some help. That's okay. Thanks, Ben. You might as well stay up here. We're going to switch it around a little bit. So first, I want to talk a little bit about myself. Everything I do is motivated by somebody I love. And today's presentation will basically be a summary of the things that have concerned me in my life and how I address them. I'm going to do this a little bit like a movie, and the entire talk is going to be a flashback. So I'm going to start with a device that we recently got FDA clearance for, FDA approval, and that is the world's first ultrasound on a chip. All of you know ultrasound as either a big cart with 6 or so mechanical probes wired by coaxial cables that cost $100,000, and there's about 400,000 of them in the room. Or more recently, people had taken that crystal technology and made it less expensive and more mobile. But historically, it's based on technology that's 60 years old. You can take a piezoelectric crystal and you can vibrate it at a fixed frequency, and you can create an echo into a body, receive that echo, and image. So I'm going to start with putting that ultrasound on a chip. We did receive the widest approval ever for an ultrasound device. We have the world's first whole body scanner. It's approved for every application. And I just want to show you that it works and then try to explain how we get to this point that we can put an ultrasound machine on a chip. So let's start with a demonstration. Now, the important thing about changing things is if you have a $100,000 machine and there's 400,000 of them in the world, there's only literally a handful of people that have the training to do that. And so what we had to do is couple putting an ultrasound on a chip with AI. And what I want to show you is that anybody can use it. And the best way to demonstrate that is to take a fresh Yale graduate, because that's anybody, who has no medical training, and in 15 minutes can do an ultrasound that a cardiologist is able to bill $1,000 for. So come on up. Let's do a demo.
Thanks, Jonathan. So I've asked my friend Amin to come up here to do a little modeling. This is the beginning of a long and prosperous modeling career, I hope.
So normally when you get an ultrasound, they match the probe to what you want to do. Our probe is, well,
I hope this works with the chest hair.
Are you going to do some fancy switching up here?
So what I'm going to do now is, so this is the Butterfly IQ. It's the first whole body scanner, as Jonathan mentioned. It takes all the functionality of a big cart of ultrasounds and puts it into a single scanner. And because the whole transducer is on a semiconductor chip, they've been able to manufacture these super cheaply for under $2,000. And so that anybody can use it, like me, there's a set of artificial intelligence tools that's going to guide me through both acquiring and interpreting a clinically relevant image. So I'm going to go ahead and show you my screen. Let's see. Just give me a second. Sorry for the AV problem. Come on.
Well, while he's getting that up, I will say something fun. So one of the nice things about making something inexpensive is it can become democratized. And I was on Twitter a week ago, and I saw the probe being used in a hospital in Kenya. And that did make me feel pretty good. Somebody brought it from Kenya. Unfortunately, it was a team from Brown.
Okay, just give me a second. This was working a second ago. Let's see. See if we can do this again. Pens Windows. Sorry, everyone. Sorry, Amin.
You know why he stopped?
It's do you want to?
I'm not an AV guy, so.
What's that? Yeah. Do you want to like?
Do you want to get it set? No.
Yeah. Yeah. Why don't we do that?
You want to start?
Do you want to go a couple more slides in, and I can try to figure it out.
How do you want to start?
Yeah.
I promise the probe works flawlessly. You can go to the Butterfly Network site. You can actually buy one if you want. And you can look at Twitter and see people are using it all over the world now. And I promise to get back to the demo. But I guess I'll distract you by talking about how my career started. So I don't know what's going on.
What's that? Yeah. This is.
Yeah. If I can recover from this, this is going to be amazing. All right, so now we're just going to go back in time. So I got my PhD at Yale, started my first company, and at the time, there wasn't very many financing options for somebody starting a company in 1993 with no experience and telling people that they wanted to mine the human genome for drugs. But luckily, I met Fred Frank, who's been a huge supporter of Yale. And true story, I told him I had a term sheet from a venture capitalist, and it had this term in it, which was known as a ratchet. Basically, if anything you say is later than you say or not as great as you say, they own more and more of your company. And if you're an entrepreneur, everything you say is an exaggeration. So basically, by the time they're finished, you know, they own your company. Fred asked me a question. He said, if you had $1 million, would you sign that term sheet? And the obvious answer was, of course not. And he says, okay, you have $1 million. He never asked me for terms. This is a true story. And a few days later, we received a check for $1 million, and we started Curagen. So thanks, Fred.
Do you want to just give me 10 seconds to try this one more time?
Really? All right.
Okay, moment of truth.
And you couldn't test it before?
It was working before. It was. Come on.
I see your phone over there.
Yeah. There's just not a lot of signal down here.
Oh, do you think maybe when everybody shut off your Wi-Fi? It probably is overloaded.
It's because it's in a.
But it was working
It's
before.
this. Yeah.
Do you want people to shut off their Wi-Fis and then?
It's not a Wi-Fi thing. It's the 4G is very weak down here. All right. Well, this doesn't seem to be. Okay. Sorry about that.
Is there any way to get a camera on his phone and then project it on the screen, the AV guys? And then we can do that, and he can just show you the screen afterwards. All right, the third time's a charm. So thanks, Fred. So I started Curigen. The idea behind the company was simple. The human genome was being sequenced. People had just released the Mosaic browser, and we were going to use informatics to mine the human genome for drugs. The company went public. Fred brought us public. Our stock went from 5 to 250. We had a market cap of $5 billion. I thought I was literally on top of the world. And on July 2nd, 1999, my son Noah was born at Yale. He was brought to the newborn intensive care unit, and he was having difficulty breathing. So I realized that everything I had done up to that date was to take a map of all of you, that human genome map that had been constructed for $3 billion publicly and $1 billion privately. The private Craig Venter project did use the scaffold from the public. It wasn't that he was 3 times more efficient. But there was a map that described all of us as Homo sapiens. And what I realized when my son was born and was having difficulty breathing is that I didn't want to know what we had in common as a species. I wanted to know precisely why my son wasn't breathing. And I wanted his genome, not a canonical map of all of us. And that night, I came up with 2 ideas to sequence DNA in a massively parallel way and to prepare DNA in a very fast way that ended up becoming an industry known as next generation sequencing. We published the work in Nature, and we showed that we could, for the first time, instead of sequence genes, so when I was here to give you a perspective in my PhD, over a 6-year period, I sequenced 1 gene, the SLIT gene, as was mentioned earlier, which is involved in neural guidance. But that gene was made out of 9,000 letters. And it took me 6 years to decode that gene. And what we developed at 454 was the ability, instead of taking 6 years to do a gene or $3 billion to do a genome, was the ability to do genomes inexpensively. And when we first used the technology, we sequenced bacterial genomes, which were about 5 million letters, so it's a lot more than 9,000, 5 million letters. And basically, some people said, this is okay, but it's not going to scale and it's going to be really tough. And other people said it was going to be big. And since there's entrepreneurs in the audience, I want to give a little guidance, and that is make sure you put the guys that say it's going to be big on your advisory board and avoid the other guys. And that's precisely what you do. On a serious note, you do want to get great advisors, and you want to get great mentors, and you want to get those people that do believe in you before things work. Afterwards, it's easy. Can I have my water? So we had a new technology, and for the first time, we could rapidly sequence whole genomes. When you have a new technology, the first thing you have to do as an entrepreneur is be an evangelist. You have to go out to the world and tell them why this is a good thing. Now it's pretty obvious. Anybody who's sick with cancer has that cancer genome sequenced and is given a specific medicine. But at the time, it wasn't obvious. So what we have to do is we have to do projects with people to show the world how powerful sequencing genomes could be and how much utility there would be from it. And so my job was to be an evangelist, call people and say, let's do a project. I'm going to quickly just go through some of these projects, but I think they're pretty interesting. So J&J had a drug molecule, but they didn't have the target for that drug molecule. And if you have a key and you don't have the lock, it's hard to develop it. And so they wanted to know where in the tuberculosis genome was this drug binding. And what we did to find out is we actually sequenced the whole genome of tuberculosis strains that were either resistant to the drug or sensitive to the drug. And these are 5 million bases each, and at the time, it normally would take years and at least $10 million to do a genome of this scale. And here we did 3 of these genomes and just compared them. And sure enough, the difference in the genome was the gene that encoded the target, and J&J was able to advance the first new tuberculosis drug in 40 years. That's the good news. Now, I started the talk by talking about my son Noah and how he reshaped the way I thought about problems in terms of going from all of us as a collection to personal genomes. But I also learned other things from Noah. And what Noah told me recently is he said, Dad, I want you to tell me all the mistakes you made so I can avoid them. So one of the things I'm going to do throughout the talk is point out when we made mistakes. So here's a case where we invented a technology, next generation sequencing. There was no software at the time, so we had to write all that software. We then took those samples from J&J, decoded those entire genomes, analyzed those genomes, found the difference, and they were able to advance that drug and get a science paper. But what we noticed was our name wasn't on the paper. And so I actually called and said, you know, we did all the work, so we were a little shocked by that. And they said, not to worry. If you ever find a paper version of this, you can look at reference 15, and they do thank us. So maybe a little lesson there. In the first case I showed you, we use sequencing to sequence a whole genome and to put together that genome and analyze that genome. Here's a case where instead of doing a genome, we want to sequence a single gene, but we want to sequence that single gene 1,000,000 times. And the reason you'd want to do that is cancer is a disease of the genome. Something goes wrong in your genome, and it goes wrong in a single cell. And so when you have a tumor, that's a population of cells, and you want to look at that same gene, potentially, in millions of those cells. And in our case, Matthew Meyerson, who is a professor at Harvard and worked at Dana-Farber, had a patient and wanted to adjust the therapy of that patient based on the genome that that patient had. And so for the first time ever, we sequenced the same gene from that patient 1,000,000 times, and sure enough, we found a break in the gene that would make the patient resistant to the drug in a small percentage of those cells, and Matthew Meyerson changed the medicine the patient was getting because of that. I was pretty excited about it, but another lesson. Our lawyers flipped out because we had sold our machine, we had done this sequencing, and we had never put on the machine it was for research use only because it wasn't FDA approved. Now, the interesting thing is, it actually took the FDA 10 years from this work until they approved DNA sequencing as a companion diagnostic. However, during those 10 years, luckily, physician scientists like Matthew Meyerson were able to go to their institutions and get an IRB and practice this. So it's a great testament to the power of physician scientists, because they were able to advance this science, make it a standard of care at all the tertiary centers long before the FDA was able to approve it. This is probably another little lesson. We wanted to show the power of the technology, and we had an ability to grab a single molecule and sequence that molecule. And one of the things that I had always wanted to do was sequence dinosaurs. And before you laugh, I really wanted to do it before the movie Jurassic Park came out and had a long-standing interest in it. And so now we had the ability to grab single molecules. So I picked up the phone and I called the world leader in ancient DNA, Svante Pääbo, at the Max Planck Institute, and said, I would like to sequence a dinosaur. He didn't hang up the phone. And I explained to him that I had seen in Science an article where they had taken amber, broken it up, and gotten DNA that was 20 million years old. And I figured if you got DNA that was 20 million years old, you could get DNA that was 65 million years old, since I had a much more powerful technology, and we should be able to sequence a Tyrannosaurus rex. When I told him this, he said, do you believe everything you read? And I said, yes. And he said, those papers were never reproduced. None of that amber stuff is real. No one has ever sequenced DNA 20 million years old. That said, you can sequence DNA that's 100,000 years old. And so upon hearing that, I said, fine, let's sequence a caveman. At that point, he explained that a graduate student will take their whole career, just like I took 6 years to decode that gene with 9,000 letters. At this time, a graduate student would take his entire career to get a femtogram of ancient DNA. So he wasn't willing to part with ancient DNA from Neanderthal. But he did say things like mammoth and cave bears, you can actually have a whole steak of them. And so he sent us DNA from cave bear. And here in Branford, Connecticut, we ran an experiment on the 454 machine. And at that moment, we had decoded more ancient DNA than had cumulatively been decoded in history. I was pretty excited. Svante said this was his dream. He had wanted to decode Neanderthal for decades, but didn't have the technology. And it was an example of perfect timing. It was literally the 150th anniversary of the find of Neanderthal in Neanderthal Valley. So Svante went to the Max Planck Institute, and they immediately gave him $5 million, and we sequenced Neanderthal. So amazing project. Also probably important to me because there was an article by Nicholas Wade in the New York Times, and it was talking about Neanderthal, and my sister called me and said, I finally understand what you do, because it was in the New York Times. But unfortunately, when I looked at the article in the New York Times, it was just about bringing back Neanderthal, because they had pretty much ignored our paper, called George Church, who's an amazing scientist, and he said, now we can bring back Neanderthal. And so that was all the article was about, bringing back Neanderthal. Nothing about understanding our origins, why Neanderthal never crossed a body of water. They couldn't see the other side. But when man came out of Africa, we went to Easter Island, and there's only 200 gene differences in a big way between us and Neanderthal, and so this might hold the key to, our higher intellect. Nope, none of that. Just bringing back Neanderthal. Oh, one other point. I do point out, someone did sequence mammoth. I felt scooped until I saw the picture that was run with the article in Nature, and I realized the two scientists that did mammoth were leaning on the machine that we had sold them. So I didn't feel as bad. I think one of the most exciting things about the technology is we could use it in a way that hadn't been used before. So instead of having a gene that you wanted to study, now you could actually literally survey things. So Ian Lipkin had a pretty interesting situation. He had gotten samples from 3 patients who had all received organs from a single donor, and they had all promptly dropped dead. And he sent us those samples, blood samples, and we literally started to just randomly decode the DNA in the blood. And what we found in all the patients that had died was a virus, and a virus that's normally non-pathogenic. But remember, when people have an organ transplant, they're taking immunosuppressive drugs. And in that case, it became lethal. And so the New England Journal called this the new age of molecular diagnostics, because now there's a simple diagnostic test you do, and before you transplant that organ, you check that they don't have this common virus, which an immunosuppressed person kills them. So that was the interesting side on the science. In terms of the popular press, this time the 454 machine was featured in a movie. So I got invited to my first premiere, took Noah along. We went to the premiere of Contagion. Sure enough, there was the machine, and it solved some incredible virus mystery. Unfortunately, it was not a box office success. So Noah said, big deal. All right. I hope by showing you that transplant case, I'm really reinforcing something you've had a discussion on, each of you, for the last 20 years, the role of nature and nurture, the role of genetics and environment. Because it was clear in that virus case that if you weren't in an immunosuppressed background, it wouldn't have been lethal. Here's another case from an environmental case. Again, Ian Lipkin was talking to researchers. They had collected material, literally jelly, from hives all across the United States. And as many of you know, these hives have been dying, and it's a huge mystery. And so again, we literally just random decoded DNA. And you could do that now because we could sequence 1 million times faster and 1 million times cheaper. And instead of sequencing, again, one gene that's 9,000 letters long, we can sequence millions of genes, put those genes in a database, and see what's different between colonies that have collapsed or are collapsing and healthy bee colonies. And sure enough, we found a virus. After it was published in Science, somebody actually took this virus and infected a hive, and it collapsed. And so by Koch's hypothesis, you would say that that's causative. But what you're forgetting is the environment again. Something's happening in the environment allowing this otherwise benign virus to collapse the hives. And so we still don't have the whole answer. Why are these hives so sensitive to this virus? I like finishing things that I start. We created this technology because I wanted to sequence my son's genome. My son had recovered. He had recovered well enough that actually yesterday he finished his last exam of his freshman year at Yale. So he's fine. But we did have to sequence a genome. And in discussion with Richard Gibbs, we thought the best genome to sequence was Jim Watson, because we didn't want to get arrested digging up Francis Crick's grave. So I visited Watson. He agreed to let us take some blood, and we sequenced his genome. We had a big public spectacle when we gave him his genome, and some reporter asked him, why did you allow Jonathan to sequence your genome? He said, well, I gave him the stuff because Watson had been involved in the original genome project, and it was this 10-year, $3 billion project, and he figured I couldn't do it, so it was safe to give it to me. But anyhow, we did decode his genome. We did put it on the internet, and it was the first of the rest of us, because now hundreds of thousands of genomes have been sequenced, and it's becoming more and more part of medicine. I do like saying the lessons, as I mentioned. So the good news was Science Magazine Breakthrough of the Year. The bad news is and Fred will remember this, I lost my job and my company was sold because the board didn't think there was much of a future in this genome sequencing business, once the fanfare was over. So I lost my company, which is a tough thing. But when you have kids, you can't let them see you down, so you got to dust yourself off and do the next thing. Noah asked me a question when I went to him and told him about the Watson genome. He asked me, or suggested to me, that if I had read the genome of Watson, wouldn't it be more advantageous to read people's minds? He thought that would be a lot more lucrative, hence why he's going to Yale now and studying economics, than sequencing genomes. And he literally had this vision of a room like this, but Bill Gates is in the room. And you figure you want to read that mind. I actually took that conversation seriously, and I thought about what we had done. And what we had done in genome sequencing is we had taken ideas from the semiconductor industry, the idea of a monolithic idea, doing something on a single substrate, and had sequenced. But we hadn't really taken the full power of that industry, which is semiconductors. And I thought maybe we could make a semiconductor that would literally be an interface between the digital world and the biological world. And all of us have seen what semiconductors can do. We saw semiconductors create a revolution in computing when somebody from Intel put a computer on a chip. All of us saw when somebody made a semiconductor that saw photons and turned them into electrons and allowing a chip to become a camera. And on my iPhone, I saw Kodak go out of business. So we've seen how semiconductors disrupt industries. The original idea behind making a chip that would see chemistry was literally to have something universal, something you could implant in someone's mind if you wanted. But I did go back to what I knew, which was DNA sequencing. And I knew that if we could see chemistry, we could see DNA sequencing. And I also knew when I had started 454, we were first. We literally had a monopoly for 2 years. We sold $100 million worth of machines in our first year because there was nobody out there that had the ability to decode DNA rapidly. So when I lost that company, and I had to start again years later, it was an industry. There was strong competitors that were well-financed. And I knew I couldn't raise enough money to compete with them. There were startups that were raising $500 million to enter the sequencing field. And so I needed a different advantage. And what I decided to do is instead of taking that idea from the semiconductor industry of the monolithic idea, I would take their entire supply chain. $3 trillion has gone into the supply chain that gives you electronics. The factories that produce the chips on these wafers now cost up to $10 billion. So I figured instead of trying to raise $500 million, I would finance the company with friends and family and leverage that supply chain, that semiconductor supply chain. And that's what we did. We made the world's first chip that would see chemistry. When we had created 454 and when the companies followed us, they all sold sequencing machines that cost $500 million. Ours cost $500 million, theirs cost $500 million. So re-entering the field wouldn't be first, so I needed an advantage. I got it from semiconductors. And I also never want to do the same thing twice. So instead of selling a $500,000 machine to 100 people, we wanted to sell a $50,000 machine to 5,000 people. And that's what we did with Ion Torrent. And it was a game changer because it was 10 times less expensive. And instead of taking a week to run an Illumina machine, you can get data in 2 hours. And that became incredibly important in both diagnostics as well as some outbreaks. So a few summers ago, there was an outbreak in Germany. 700 people were rushed to the intensive care units. 50 of them died. And it was the ion machine on a $99 chip in 2 hours that decoded a superbug that was killing those patients, allowed people quickly to make a diagnostic, and screen food for this superbug. So I was able to re-enter the field. A lot of people have observed that DNA sequencing advances are going faster than Moore's law. What I want to show here is we're actually not going faster than Moore's law. We're just catching up to the densities that Moore's law has achieved. So when we invented the 454, the first high-throughput sequencing, you could sequence 4 strands of DNA, 4 pieces of DNA on the end of a human hair. When we did ion torrent, we were at a density, and we were using a factory that made chips from the 1990s to decode that bacterial genome, that 5 million letters, and you could sequence 400 pieces of DNA on the space of the end of a human hair. Now, we wanted to sequence whole humans, and we wanted to do it for $1,000. So the trick there was just going to a newer foundry, these $10 billion factories that make these wafers, because if you look at anything you've bought electronic, any chip you've bought, if you compare a 10-year time span, you'll realize that it's 1,000 times more powerful. And that's Moore's law, of course, and that's perfect. So we actually sequenced Gordon Moore's genome on the ion machines, because if you remember when I first showed DNA sequencing, the skeptics said it would never scale. You wouldn't be able to do bigger and bigger genomes. So to cut them off, we sequenced Gordon Moore's genome, the father of scaling. But the point was that now you could do a $1,000 genome because a human genome is 1,000 times bigger than a bacteria, and you just go to a density that gives you that 1,000-fold improvement. So we're just catching up. And this is important. It means that once you've caught up to Moore's law, where those factories are now, you're not going to keep on seeing these 10 or 100-fold advancements because you have other bottlenecks. You have to have the compute to handle it, and you have to face where you are in the development of Moore's law. So biologists weren't better than electronic guys. We were just catching up to them. I think every section I have to have a lesson from it. So in this case, we had a new machine, the ion machine. It was featured on a website and in the Jurassic World premiere. And the good news was this was the most successful movie premiere up to date. So Noah said, OK, you're vindicated. That was the good news. The bad news, instead of calling it the ion machine, they called it the Hammond 20. Kind of disappointed with that. But we did have a blockbuster finally. This time I sold ion. When 454 was sold, it wasn't by choice. I think Mark Twain probably has some quotes about that. So it wasn't by choice. And I had always felt, and I probably had talked to Fred about it, but probably nobody else, that the guys that bought 454 had got it 20 cents on the dollar. But you can't say anything publicly because people will say, yeah, sour grapes or whatever. So I didn't say anything to anybody. But when we went to sell ion, I sold it for exactly 5 times more. So it would be $1 on the dollar. So we sold it for $725 million. And that was good. And we had shown that semiconductors could affect computing, semiconductors had affect imaging, and now semiconductors could really change the way DNA was sequenced. So what's next? In the beginning, I told you, are we going to get that demo? There's just no way. Can we do the demo and somehow people see on the screen or get a camera? There's nobody who can get a camera on it. Okay, but after they can do a video of it? Did you find a good one at least? All right. All right, so if you want to see the live demo, send me your resumes and we'll hire you. So I said in the beginning that everything I do is driven by people I love. And I like this quote by William Gibson. I was funding some research into non-invasive surgery. That's when you use this high-intensity focused ultrasound to intersect inside the human body and do surgery without opening the body. And I was funding it in an orphan disease called tuberous sclerosis to get rid of growths in the kidney. And I really felt that I was living the William Gibson's quote, because here was a center, and there's about 10 of them or so in the United States, with 10 million plus equipment. So it's too expensive, it was too slow, and it wasn't precise enough. But it was the future. And about that time, I met a physicist, Max Tegmark. And Max was giving a talk on how to image the universe. And he had worked out a way to use thousands of individual antennas to create a single image. He had worked out some math problems. And one of those math problems involved something called the butterfly network. And I approached Max and I said, I want a way to image inside the body and ultimately do non-invasive surgery in the body by using tens of thousands of micro-machines on a semiconductor chip. I had just been successful sequencing DNA with a semiconductor chip, and I wanted to use a semiconductor chip to be able to image my daughter at home instead of going into a hospital. And I thought he had an algorithm that would enable it. And I said to Max that I'd love to start a company with him, but I think it's going to be a tough problem, and I'd like the smartest kid from MIT to start it with us. And he said, that's no problem, because Max always put himself on the admissions committee at MIT and would look for kids that he knew were fantastic so he could steal them immediately and bring them to his lab. So he introduced me to Nevada Sanchez, who had just won a Hertz Fellowship for his PhD at Stanford. And we convinced Nevada to take a one-year sabbatic 5 years ago. And told him we wanted to put an ultrasound on a chip. I brought in Keith Fife, the best chip designer I know, and probably one of the best chip designers in the world. And we did it. We put an entire ultrasound on a chip. So instead of having a $100,000 device, you can go to our website and buy a $2,000 device. The Gates Foundation has approached us, so we will be making a sub-$1,000 one for developing countries. And this is basically how it works. You have the chip on the front of a probe, and I'm wrapping up. And it works by having these micro machines on a semiconductor chip. So now, instead of having specialized factories that wire together ultrasound machines and having 6 probes and costing $100,000, you make them on these wafers, and you have 9,000 transducers. And instead of having a fixed crystal for every frequency, it's universal. We did get the first and widest clearance ever by the FDA for a micro machine driven ultrasound. And the interesting thing is it does work as well. Right now is a $60,000 machine with 3 to 6 probes, but we're literally just getting started. We have AI to help anybody guide it, and we have AI to do the analysis. You can do an ejection fraction with no training. My son did it with literally 5 minutes of learning how to do the machine. And the beautiful thing about this is it's a virtuous circle. Every time the machine's used, if a cardiologist doesn't like that ejection fraction and changes the volume of that ventricle, the machine sees it, learns, and gets better. And so the machine is not only getting better at heart, it can now do AAA, and it's literally learning with every machine that's out there. And it was exciting for me. I literally found this on Twitter. Somebody took our machine to Kenya, and what's exciting is that they were doing things that they had never done before with these patients. And it matters because 2/3 of the world have no access to imaging. Really, since antibiotics, most medical breakthroughs have not been democratized. There are 40 million people in the healthcare profession that absolutely want this window into the human body, and less than 1% of them have access. So we're super excited about that. And I think we've really shown that you can really take this concept of semiconductors and apply it to other fields. And so I do have an incubator. The incubator starts our own companies. They all have the exact same business plan. We make a device no one has ever made before. We put that information in the cloud. We use this revolution in AI to create a virtuous circle about it. And if you write the jobs at for Catalyzer, love you to join us. We have locations in Guilford, Connecticut, New York City, and Palo Alto. And what's really important when you're talking to students who are starting their career and want to do good science and want to build their career, you can do that at a company. We've gotten the cover of Science, Nature, at our startups, and you can get recognized. So this is Nevada Sanchez in his 5th year of his 1-year sabbatic from Stanford, and Forbes 30 under 30. I'll end with an ego shot. I hope that's okay. I like it when there's more students. I was fired, and interestingly, almost to the day, 10 years to the day that I lost my company and I was fired and I was bummed out, Obama gave me a medal for the exact same thing I got fired for. So I thought that was pretty interesting. Thanks. Thanks.
Sorry on the demo.
Are you going to show the clip? Can we show a quick clip? Will it take 30
Is it
seconds?
30 seconds? Yeah, 30 seconds. Promised.
This is my vindication.
The problem wasn't ours, we'll blame somebody else. Communications between the device and the screens.
Do we have the AV guys? Can we switch over to the other screen? Okay.
I think it's butterflynetwork.com.
Anyways, it's okay. We won't try to do this again. All right, sorry for the AV troubles.